and engineering, and is director of the Rosen Bioengineering Center here at Caltech. And she developed directed evolution in chemistry and received the 2018 Nobel Prize in chemistry for her work. Francis.
Wonderful. Great. If we could pull up my slides, I'd appreciate it. Greetings, everyone, and thank you, Anima, for this really exciting overview of what AI can do in science. If we could bring up my slides, please. I love proteins. This is the group that does all the work. I always start with that to remind us this is the work of the young people whose ideas are so important for bridging AI and science. Now, I love these proteins and particularly the enzymes, because you imagine the enzymes are what are responsible for taking renewable resources like carbon dioxide, sunlight, and turning that into complex biological things like a tree or you and me. That chemistry is absolutely amazing. It's clean, it's recyclable, it's renewable, it's efficient. And to me, that is the best chemist on the planet. So I started off wanting to engineer new versions of these molecules, not just to do the chemistry that nature has invented, but actually to replace chemistry that humans have invented because we're not nearly as good at it as the enzymes are. So imagine if you could genetically encode all of chemistry, basically replace the chemists with microbes. It makes me very popular in chemistry audiences. But if we could do that, we could get many of these fabulous properties of enzymes into our circular economy. All right, so that's the dream. In order to implement it, what we have to learn how to do is compose new DNA. Rewrite the code of life to give us enzymes that are better than what nature gives us at our jobs. And of course, no one knew how to do that. When I started out at the beginning of the DNA revolution, that was a really hard problem. And I'll just tell you, it's equally hard today. We've made very little progress in physics-based models of the mapping between the DNA sequence and the function that we want in biology. So that led me to use the most powerful design process that was ever invented, and that's called evolution. And by evolution gives, I guess my slides aren't working here. This is a lovely little animation. Oh, there we go. You're turning the crank this is the first algorithmic design process, right? You're turning the crank of mutation and natural selection that's given rise to all the diversity of the biological world. Works at molecule scale all the way to ecosystems. Imagine that. Engineers in the audience, eat your hearts out, because here's one design algorithm that works at all scales. And we humans have not figured out that process, but the biological world did. So it made sense to use this process because evolution has not stopped, right? Why not breed molecules like you can breed cats and dogs? Humans figured that out a long time ago, how to use evolution to move into the future. Now we can do it at the level of molecules. So now I want you to imagine what you're doing is you're freeing yourselves from the constraints of being biologically relevant. Biologists in the audience, I'll now offend you. You study the tiniest fraction of the space of all possible biologies and all possible proteins. And now, if we free ourselves from the constraints of having to serve biology, we can go out and explore a universe of protein possibilities where I claim lie the solution to the energy crisis, the cure to cancer, and maybe even the cure to death and taxes. But you have to find it. You have to figure out what DNA will encode that. And it's a mighty big space. So I always show this because I want If those of you who have read Jorge Luis Borges' fabulous story, The Library of Babel, understand the enormous dimensionality and the enormous size of this space of all possible proteins. Figure it's 300 amino acids long. That's a really big number, 20 to the 300th power. It's bigger than the US national debt, you know. So, and even if you change 1% of a protein sequence, you're already up there in the many billions of possibilities. So no experimental method, no empirical method like evolution can search that. Now, in Borges' story, the librarians would throw themselves off the balconies in despair of ever finding a single meaningful book. This is the library of all possible books. In my library of all possible proteins, it's equally problematic. Most of the sequences, the ways you can put amino acids together are total gibberish. Yet evolution works. And this was the great insight of John Maynard Smith, who also wrote a wonderful short story in 1970, published, where he said, okay, we've got this space where most of it's gibberish, but evolution works. And that tells me something of how these DNA sequences have to be arranged, how these protein sequences are arranged as functional proteins in the space. And he basically made the argument that for evolution to work, you have to be able to walk around in this space of this gene space, one step at a time. Because if you make multiple steps, right, the probability of function just drops to zero. So evolution works by walking around in this space, one step at a time. And that means that you could implement a very simple process for directing that evolution. So we think of this as an optimization problem, and this was many years ago, where if the landscape is smooth, any fool can optimize. If it's rugged, on the other hand, you take a step, a mutation, and you fall into a crevasse of non-function and evolution stops. But the fact that we're here to talk about it today means evolution has not stopped. And to me, it told me that the landscape on which proteins evolve, at least, has to be more smooth in some of its many dimensions. All right, so that insight means you could implement a very simple optimization algorithm. Any fool can optimize on such a landscape where you take something that nature has given you, for example, the work of 4 billion years, a parent enzyme that you can literally scrape off the bottom of your shoe. You make a few changes randomly to that, and then we could use the tools of recombinant DNA to put that new DNA into microbes. They would express the proteins. And then you, as the breeder of molecules, decide who goes on to parent the next generation and who gets thrown in the trash can. And that process really works. You just optimize then one step at a time. You take a natural enzyme, ask it to do something new. It's generally less enthusiastic about that. You're now on this red fitness landscape, but you turn the crank of mutation, artificial selection now, and you can re-optimize for the novel function. And in fact, I got tenure, so that must mean it works really quickly. And in fact, it works so well that thousands of labs in everyday products that you use, from your laundry detergents to your pharmaceuticals, are manufactured using enzymes that have been optimized by directed evolution. I mean, your laundry detergent, what self-respecting enzyme wants to work in your laundry machine? Believe me, they don't. Natural enzymes don't do that with surfactants and bleach, but these are all optimized to take stains off of clothes in your laundry machine. So here's a process that works for all sorts of different problems. And of course, I got the biggest accolade of my life as a result of that when I was asked to go down the hall, go down the road to Warner Brothers Studios and appear on the Big Bang Theory. And for the maybe 2 or 3 people on the planet who haven't heard of this TV show, it's about nerds at Caltech who, after yeah, who are endearing physics graduate students, but after 12 seasons become extremely annoying professors who are lobbying for Nobel Prizes. So I got to show up with Kip Thorne, you could see, and George Smoot. We had a very good time uh, being lobbied for. I got to throw Sheldon's oatmeal raisin cookies in the trash. In any case, that was a fun thing. But all right, let's get back to science. What did I learn? Proteins evolve this way. They can be optimized this way. New functions come quickly. Small changes in the sequence give rise to measurable and important changes in function. And anybody who studies antibiotic resistance or viral evolution knows this very well. Here's the scary fact, though, and this is why it's so hard to design enzymes, that mutations, very small changes in the protein sequence, 20, 30 angstroms away from the active site, give rise to big changes in catalysis. And we can't even explain those, much less predict them. All right, so that's our problem. And we nonetheless have this algorithmic process for optimization. So this is 35 years ago. We knew this 35 years ago. But recently, in the last 10 years, I decided to take on a more difficult, what I thought was a more difficult problem, which is how do you produce novelty by evolution? How do you do something that you cannot go to a database and find a starting enzyme for, right? Because to start the process, you have to have some measurable level of that activity, and then you turn the crank. But if you are looking at a function that doesn't exist in biology, at least as far as we know, nobody's found it yet, you cannot go and find a starting point. And it would annoy the hell out of me because people like David Baker, who won the Nobel Prize last year for AI and enzymes, would start turning out terrible enzymes. They were awful, but they could do things, for example, that you couldn't go to a database and find. So I said, oh, that just annoys me. And I said, surely evolution can do this and do it better than AI. So, uh, how do you get out there and find this novelty? And I stewed for several years until I realized I was thinking about it all wrong. Because if you study evolution in nature, you realize that new enzymes are being made all the time. Nature generates novelty all the time. It's just maybe you didn't catch it, but sometimes we can catch it in action. And what we realize is that novelty, where does it come from? It comes because it's already there. The biological world is so diverse that there's already enzymes out there whose biological function might be one thing, but given a change in the environment or change in opportunity, it can do something else. It's like a graduate student, right? So evolved for one function, but has multiple capabilities that can be drawn out by evolution when the opportunity arises. So if you come to this realization, then you can just apply the same process and create novelty by recognizing the power of diversity. So my favorite example, I want to open people's minds. So I said, okay, let's make carbon silicon bonds because no one has ever found organic molecules that incorporate silicon in biology. Humans make it all the time. We have 50 products in this room that have carbon silicon bonds, silicones and earbuds and hair gels and stuff like that. So let's go ahead and make an enzyme that can do that. And that means using chemical intuition to create something that we know chemically might have that capability, but finding an enzyme in nature that already has that. So, you could start thinking algorithmically here. So if you realize that enzymes can activate oxygen, a chemist might guess that it's not, if you squint really hard, that activated oxygen looks like an activated carbene. And if an enzyme can make that activated carbene and insert that into another silicon containing molecule, it could possibly carry out this chemistry. And in fact, we found lots of proteins from nature that have the ability to make carbon silicon bonds already there. It was just never biologically relevant, but they're perfectly capable of doing it. And I love this little itty bitty protein from a hot, salty pool in Iceland that does it as well as a human. This was as good as any human chemist had ever done for this reaction. 40 times it'll catalyze the reaction. It's not useful, but it's there. And I love this one because this protein, by virtue of its origin, is so stable that you can give it to a chemist and they won't even know it's an enzyme. You can make it into a powder and you say, here's a magic powder for making carbon silicon bonds, and they can use it just like any other reagent. Now, people complain and say, well, I don't know that this protein could do this. And in fact, not only does it do this, but it evolves. So you find a little, now you're starting to get the idea here, you find a little bit of activity for what you want, and you can turn the crank and make it so good that you can get it published in a snooty journal. So we published this in 2016 and the news went all over the world. No one read the paper. They only read this article, uh, which, which Bob Service had written about it. And you recognize the Horta episode on Star Trek. I have realized now through my experience that the best way to get your science out there is to tie it to science fiction, because nobody reads your boring chemistry paper, but they love this sort of stuff. So the news went around the world and mutated as it went. But the bottom line here is that once you create novelty in biology, this is a very special place where evolution works. Chemistry is going to be much harder, material science is hard, but biology is very special because once you create novelty, it opens up the route for yet more novelty. So we've made hundreds of novel enzymes by this process. But it's an awful process. So all this wonderful stuff, I got the Nobel Prize for it, thousands of labs use it, but I tell you, thousands of labs hate it because it's really painful. You have to search through all these proteins and make thousands of measurements, and you're lucky if you collect a few mutations, and you might wander around through this rugged landscape because it is rugged. It's not smooth. It's mostly empty, and you might be walking around for a long time before you end up stuck on a local optimum. So it has its big flaws. And now I get into where machine learning and AI can help. So in the old days, we would take all the data. So we were doing high throughput screening 30 years ago and throwing most of the data away, right? We would take the best ones forward and throw everything else away. And now we can use those data to build machine learning models. So already starting in 2012, so this was long before the AI craze, we were building machine learning models with Bayesian optimization to figure out what are the ways that we can incorporate the data to tell us where to go in the next generations. And that basically what we're trying to do is recognizing that the landscape looks more like this when you go out and scale. And how do you, instead of getting stuck on a local optimum, how can you discover some of the more interesting nearby peaks? So we learn that mapping by taking the labeled data into a machine learning model. And that means you measure the sequence as well as the fitness. And then, of course, as the models get more sophisticated, you can start incorporating other information into those. And so Bruce Whitman and Kevin Yang, who got their PhDs and now are at Microsoft, and both at Microsoft now, are building an ecosystem for a lot of this modeling. Now, I'll just tell you a little bit about what we're doing most recently. Jason Yang, I believe he's in the audience, developed an active learning implementation of this, because it's very much like directed evolution, only now we're updating the model as we go through the generations. And his most recent work, he can use basically, he can have uncertainty quantification and then pick pick the right mutations using an acquisition function. And I thought it would be fun just to give you one little example, sort of to concretize the implementation of this. Because what happens in an enzyme active site is a lot of times the residues in the active site, when you're starting to create a new function, have to be remodeled. And those are what we call epistatic. Basically, they're strongly interacting with one another. So that idea that you can walk smoothly up a hill falls apart when you're looking at an enzyme active site. So if you take a design problem where you're trying to get a new function out of an enzyme, and here we're trying to make one out of two possible products that the enzyme can make, that's going to require fine-tuning of the active site. And so if you, for example, find an optimal set of mutations on 5 sites, you've got a big design space, much bigger than we can easily measure experimentally. 3.5 or 3.2 million ways to do that. And what we want to do is both increase the activity and get selectivity for product 1. So that's, and if you did directed evolution, basically most of the time you'd get stuck on a very uninteresting place. And so with the active learning, what Jason and Ravi Lal were able to do, you start off, you make your set of all possible mutants, and you can randomly select some subset to measure, a very small subset to measure. And that can be done using predictors or just at random. And what you can see is in this case, they had a bunch of proteins that were what you're looking at here is if it's fully active, so it uses up all the substrate, it'll lie somewhere on this dotted line. And if it's 100% selective for the number 1 product, this is the product we wanted, you would sit here. So most of them are only selective for the product we don't want. There are a few that are reaching out into this space. So we get some information from that. Apply the active learning model now in the first generation, and you can see there's already the population has moved way over in this direction. You're getting some very good ones. And what I find really interesting about this is that the solutions are looking quite different. These are the sequences, right? SAFRI, that's the best one in the first generation. But as you move out, the best one now looks very different. And maybe you have the Y at the end. So single mutations are not, the smooth pathway is not happening here. And then by the time you go to the second generation, you're already out with really good solutions, 99% yield, and you've got very high product selectivity. And you've only measured a few hundred things. So this is the power of these machine learning models. They get you to very interesting sequences that are not obvious from the original data, but that give very nice solutions. And this is exactly what we want to be able to do the optimization in as minimal number of tests as possible. Now comes, if you've got this wonderful algorithm for optimization, that remains only one place left that's weak, and that's where do you start? And that's where I was complaining about David Baker, because what does he do? He gives us a place to start, and we can now use generative AI in order to create those starting places. So I'll just point out, Anima mentioned that we're collaborating with Argonne, and you'll hear from Arvind. I don't know if he probably won't talk about our collaboration together, using language models in order to create new enzymes. But the limitation of that, of course, is that learns from evolution. And if you're creating novelty, that means your new function is not in the data set that you're using for training. And it turns out that it's really hard from these language models to get proteins that are truly novel in terms of their functions. So instead, there's a little contingent in my lab at Caltech with Jared, I believe Jared is here, and Cheng-Hao Liu, who are developing a whole new sequence structure model where they've actually succeeded in combining continuous diffusion as well as discrete diffusion for the sequence part. So continuous diffusion for the structure part, then they're doing the sequence part, simultaneously with discrete discrete diffusion. And I don't have time to go into details, but they're both here if anybody wants to talk to them. And what can we do with this? We can start using structure-guided design to build new proteins around what we understand from the chemistry, which is, say, the structure of a carbene intermediate. And that way they can start building new protein designs around that. And these, the predictions look pretty much like what AlphaFold would tell you. And what are they doing? They're actually putting the ligands in the substrates in the structure. They're putting in the heme as well. So these are pretty sophisticated. They can do it for carbenes. They can do it for nitrines. I don't know if there are any chemists in the audience, but that's pretty cool chemistry because you can now make carbon-nitrogen bonds, not just carbon X bonds, but new nitrogen bonds. And the bottom line is this really works. This really works because for an easy reaction, you're getting a very high fraction of things that have activity. So this is your negative controls are this dark blue, and anything that's lighter than that is showing measurable activity. And so for a very easy reaction, you get a large fraction of the designs. This is a 96-well plate where you're testing each one. And so you've got all what you're making are proteins that come from very different sequences, very different structures. You're exploring this universe, this enormous universe of new proteins that you can't get to by single-step evolution. And yet many of them are functional. And they're functional at a level that would allow them to be optimized by directed evolution. So this is a way to be able to genetically encode all of chemistry. Because if you know enough about the reactivity that you can get a little bit of activity, that gives you the starting point. And so Jason wrote a very nice review very recently laying out this idea that we should be able to sit down and encode all of chemistry if you have, these foundation models basically on the broad public databases, but then have this protein engineering part that does the fine tuning. So you can steer and fine tune even these generative models. So we envision that a lot of the work will be able to be done using these models. And I'll just end with this because back in 2022, I said, because all these steps are automatable. That's the other thing you need to understand. Everything that I showed you in the directed evolution process can be automated relatively easily. That given, then, you can imagine that the whole process can be done at the push of a button. So I said in 2022, no more of this awful directed evolution. It's time for machine learning to take over. And Jason came up with this. This was in the early days of image generation. So that's the graduate student, now pressing the button. Now, you'll notice that that was on April Fool's Day that I posted that, and I'll change my mind tomorrow. But we're there. We're almost there. And I can smell it coming that we'll be able to do this. And that means, that's why I'm really excited. That means that we will be able to genetically encode all of these wonderful transformations and get bacteria who will work for sugar in order to do our chemistry. So thank you very much.
So we have time for a couple.
So you were talking about using generative AI at the beginning to get started, but then it sounded like it was still kind of directed evolution, technically doing its thing afterwards. It seems like there are also opportunities to spice it up.
Absolutely, absolutely. And that's Jason's view, is that we can actually integrate all of this. So that's where it's going. And I totally agree with you. Great.
Okay, well, thank you very much.